Hello, today's lesson is about the specific immune response. This is going to be your second activity, page 140. Let us first remember that the immune response is the battle that takes place between the immune cells and the pathogens or any non-self that tries to invade our bodies. The immune response is either non-specific or specific. As we said before, the non-specific immune response is carried out by the phagocytes, and it is not specific against a particular pathogenic agent. So any agent that enters to the body, any cell which is said to be a non-self is going to be or can be eliminated by the non-specific immune response. On the contrary, the specific immune response is an immune response which is targeted against a particular antigen based on its identity. So usually the first line of defense after the pathogenic agent finds a way to the inside of the body, the first line of defense is going to be the non-specific immune response. When this defense fails to eliminate the intruder, the specific immune response is gonna start. Remember that the non-specific immune response is also called an innate immune response, while the specific immune response is also known as adaptive immune response or acquired immune response. The cells that are responsible for carrying out the specific immune response are the B lymphocytes and the T lymphocytes. Usually, as we said before, it is a highly specific response against a particular precise pathogen and not against any type. It creates an immunological memory. This is very important, and this makes a big difference between the non-specific and the specific immune response. In the non-specific immune response, there will be no formation of any immunological memory, while regarding the specific immune response, the characteristic which makes the specific immune response a very important immune response is that it creates an immunological memory. We are going to discuss this later in this slideshow. Let's take a look into this document. You have this document in this activity in the book. This is a hen or a chicken, and it is injected with killed salmonella. Because the salmonella is a type of bacteria that is used here is killed, we call it a toxoid. If it was living, then it will be known as a toxin. So toxoid is a killed salmonella, killed toxin, or attenuated. We can also use an attenuated or weakened one, but in this case they have used or they injected the hen with a killed salmonella. 10 days later, the same hen was injected this time with a living salmonella. It's not killed. Obviously, the hen remained alive. So what happened here is that when we injected the killed salmonella to the hen, before injecting it 10 days later with the living salmonella, the hen now is introduced to the same toxin. So it's the same actually antigen of the toxin, but in this time, because it was killed, so it can do no damage to the body of the hen. But what it has done is that it introduced the immune system to the antigen of the salmonella. So the body of this hen has recognized this antigen as non-self. It developed a specific immune response to get rid of this killed salmonella. Even if it's killed, the body doesn't care actually. Any non-self has to be eliminated no matter if it is going to harm or not. So the body in this case of the hen has eliminated the killed salmonella. It has undergone a primary immune response to kill it. We call it primary because it was the first time dealing with this kind of antigens. And it has created immune cells, and this is the immunological memory we were discussing. It has created an immunological memory, or the immune cells were formed. And after 10 days, after we injected this hen with the living salmonella, these immune cells that have been developed from or upon the first contact with the salmonella, they recognize the living salmonella in a very fast way if we compare it to the primary response. So this response here is said to be the secondary response because it's the second time the hen is being exposed to the same antigen. And due to these immunological memory cells, or the memory cells, the salmonella has undergone a secondary immune response which was fast, which was enough to leave the hen alive. The same thing has happened when we injected the hen with the killed Vibrio cholera. It's another type of pathogenic agents. Before we injected with the living Vibrio cholera. So we have introduced it to the toxoid. It has formed the immunological memory, which helped this hen to resist the living Vibrio cholera to remain alive. In the third experiment, what we have done is we also injected the hen with the killed salmonella, but 10 days later, we injected this hen with the living Vibrio cholera. In this case, the hen, as you can see, was dead. So, what we did here is we injected the hen with the killed salmonella. So it has developed a primary response against salmonella. So it has memory cells against what? Salmonella. But 10 days later, we have injected it with another kind of pathogenic agent, which is called the Vibrio cholera. So the hen died. This is obviously because the immunological memory that has been formed at the level of the hen was targeted against the salmonella and not against the Vibrio cholera. And this is why we say that these cells, or the cells that are involved in this immunity carried out against salmonella and Vibrio cholera, is specific. If you take a look here into the fourth experiment, where we injected the hen with the killed Vibrio cholera before injecting it with the live salmonella, also this hen has died. Obviously, this is because in this case, the hen was immunized against Vibrio cholera. It has developed memory cells against Vibrio cholera and not against salmonella. So for it to develop the immunity against salmonella in this case, it requires around 7 to 10 days, and in 7 to 10 days the salmonella wouldn't be waiting the hen to resist it. It's actually gonna do the damage and get to kill this hen. So this is a very important document that shows that the specific immune response is said to be specific because it's targeted against a particular antigen only, and after the primary immune response, the body will be immunized, will form memory cells, which are long-living cells. We call them memory because their job is to remember this particular antigen if the body met this antigen again. So make sure you study this document really well. So the immune response developed 10 days after the injection of salmonella or Vibrio cholera is specific. Let's take a look into this important document as well. If we injected this hen with an aged Maltosida, which is also another type of pathogenic agent, when we say aged, we are meaning that it is a toxoid as well, a weakened or attenuated toxin. So it is a toxoid. Of course, because it is aged, it's not gonna do any damage to the hen, and therefore the hen is gonna survive. It's not gonna show any disease, and it's not gonna die. If we use this same hen, which is now immunized against this particular pathogenic agent, which is the Maltosida, if we used it and we injected it, but this time with the fresh Maltosida, you can notice that no disease or death has occurred. Okay, so this is because it is now immunized against the Maltosida, because the antigen of the Maltosida has been introduced to the immune system of this hen, and it is now ready to form a very fast immune response because of the presence of the memory cells which were formed upon the first contact with this Maltosida. Now this same hen, as you can notice, didn't die when we injected it with the fresh Maltosida. Now, if you get another hen which was not immunized, it wasn't injected with the aged Maltosida, but directly was injected with the fresh Maltosida, as you can see, this hen is gonna die. So the difference between this hen and this hen, they were both actually injected with the same fresh Maltosida, but this hen was immunized against it because few days later it was injected with the toxoid of this Maltosida. So it has formed the memory cells that will help it remember this Maltosida when it ever enters its body again. And this is what happened in this case, and this is why it didn't die. While this hen was not injected with the aged Maltosida, so it didn't form any memory cells, and this is what led to the death of this hen. Remember that a toxoid is an attenuated or weakened toxin. It can also be killed toxin. Once introduced into the body, this toxoid, it doesn't harm the body itself, but still the body will form a specific immune response to eliminate it, to eliminate this toxoid, and the body will develop an immunological memory which will remember the same toxin if it accidentally entered the body again, and it will cause, this is gonna cause, due to the presence of the memory cells, the secondary immune response, of course, is gonna be faster than the primary one. Take a look, please, into this document. This document is giving us a summary on the types of the specific immune response. When we talk about specific immune response, we have two types: the humoral immune response, the humoral specific immune response, and the cell-mediated immune response. The cell-mediated immune response, it's carried out by the T8 lymphocytes. As we said before, the T lymphocytes are of two types: the T helper, which are characterized by having CD4 or cluster of differentiation of type 4, and the T8, which are characterized by having the CD8 on their surface. The CD4 cells, the T4 I mean cells, are responsible for secreting cytokines, which are said to be interleukins. These interleukins are of two types: the interleukins of type 4, which will activate the B lymphocytes to secrete antibodies to get rid of the antigens. And as we said before, the B lymphocytes ready to start secreting antibodies are said to be plasma cells. And the interleukins of type 2 secreted by the T4 cells will activate the T8 lymphocytes, which will undergo the process of lysis to the infected body cell, and this is gonna kill and eliminate the infected body cell. Specific immune response, as we said, is the humoral or cell-mediated. You're supposed to determine and distinguish between these two types. Humoral is the one carried out by the antibodies, which are secreted by the B lymphocytes after they are activated by the interleukins of type 4 that are secreted by the T4 lymphocytes. The cell-mediated specific immune response, it's carried out by the T8 lymphocytes, causing the lysis of the cell after being activated by interleukins of type 2, which are secreted by the T4 lymphocytes as well. Take a look into the humoral and the cellular immunity. The humoral immunity is actually carried out by the antibodies that are secreted by the B lymphocytes in a way that these antibodies are going to attach to the antigens, and the antibody and antigen complexes will be eliminated by the phagocytes. So the cooperation takes place between the non-specific immune response and the specific immune response, because after the specific immune response, in this case a humoral specific immune response is taking place, the phagocytes are going to eliminate the complexes formed from the antigen and antibodies. As for the cellular immunity or the cell-mediated specific immune response, this immune response is carried out by the T8 lymphocytes that are going to attach to the infected body cells, causing the lysis. And of course, you shall remember that when we talk about these two types of specific immunity or specific mediated immune responses, cell mediated and the antibody or humoral mediated immune responses, keep in mind that the T lymphocytes, particularly the T helper cells, are the ones that are going to start both responses, cell mediated and humoral mediated. This is because when the T helper cells secrete interleukins of type 2, they are going to activate the cytotoxic killer cells, which are the T8 cells, to carry out the cell-mediated immunity by lysing the infected cells. And when these T helper cells secrete interleukins of type 4, they will activate the B cells, which will change into plasma cells to secrete antibodies and to carry out the humoral mediated immune response. Thank you very much. This activity is very important. Make sure you study it very well. Bye.